Welcome, everybody. My name is Steve Drucker. I'm from the Department of Chemistry and Biochemistry. It's my great pleasure to introduce my colleague and friend, Sudeep Bhattacharyay. He's a prolific researcher in our department. He's professor in chemistry and biochemistry. His accomplishments are very long list, but I will say briefly, he's a superhero. That will summarize it, because he masterminded the acquisition of our blue gold supercomputing cluster, which is a really big deal. And I was just chatting with someone at a collaborator at a small college, fancy liberal arts college, and I know this person, and they're looking to set up a cluster. And they were blown away by what we have as a public comprehensive university. And this is attributable to Sudeep's efforts. And he got this thing started from a closet. It's a couple of little, like an abacus or something in a closet, nothing very fancy. He built it up, and now it's a supercomputer thing. So anyway, that's the thing that you should know. It's not on the bio. And his research, when he's not masterminding high performance computing system, his research is computational biophysical chemistry. It's a lot. There's 4 things. Computational biophysical chemistry. And today he's going to tell you about some of his work with
COVID-19
in his research group with his collaborators and students on breakthroughs in understanding COVID-19.
Well, thanks, Steve, for the presentation. I'm a little bit pleasured by the supercomputer, which is, the effort by many of us, including yourself, for contributing the overall acquisition of the cluster. So thank you to everyone. I'll start. Today I'm going to talk about our research on COVID-19. The chemistry angle is there, but most importantly, I'm going to share how our undergraduate students worked during the time of that COVID-19 lockdown period, when we're kind of out of luck almost in everything. We're not able to access our library. We're not able to access the experimental lab. We try to do science and learning, continue to do that during that time. And I think I'm going to share that, and I'll be pleased to do that right now. Overall, the talk is outlined into 5 sections. I'll introduce something very basics. Most of you know what is protein, COVID-19. I'll talk about oxidative stress, and then lay out an objective of what we wanted to study. I'll briefly discuss about methods and results, and give you a concluding summary. So proteins, as you probably see, these are different color spheres. You can think about a necklace comprising of different beads of different colors. In a way, we can say that essentially they are a sequence of different amino acids. So if you carefully look at each of these beads, are different colors as well as different atoms in it. And these amino acids, which are called amino acids because they have a NH2 group and a COH group. They're called amino and carboxyl group. Now, they are the workhorses of life. All the life forms in this kingdom of living being, they are made up of 20 amino acids. They're coded in our genes, and surprisingly, if I take you to compare ourselves with this fruit fly, we as a human being are 60% similar to this fruit fly. Kind of surprising, right? Not surprising to the biologists. They talk about all this time. Now, proteins are immensely important for all life forms. As I said, they are used in transport, like storage. Our hemoglobin that transports oxygen in our blood is made up of protein. The antibody that saves us from microbes, enzymes that do millions of catalytic reactions in our body, as well as messengers, and finally the structure, big muscles, and we all know that, made up of proteins. Viruses, sometimes not called as a living form, they need a host cell to replicate, and they are having a core where the genetic material is stored, but they cannot really replicate unless they get into a living host, and they replicate only inside the host. And they also have proteins, like these are proteins that are popping out of this core shell in which the genetic material is preserved. Our lab looks at proteins, and our focus is how proteins motion makes all these dramatic effects into reality. What I mean, that look at this is from our recent simulation in Chem 406 biophysical chemistry. These shape-shifting kind of cluster of atoms, why and how they're connected to catalysis, how they're connected to electron transfer, how they fold even within a cell, and the viral proteins, as well as drug designs. If you know, drugs are small molecules, organic molecules that can go inside this cell, get stuck on a particular protein and stops it from functioning its original action. So this computational part of our lab is having three of these projects: drug design, as I said, a simulation-based approach to look into how proteins and enzymes function in a crowded environment. And we also know that viral proteins and how its dynamics play a role in oxidative stress, and that's what I'm going to kind of talk about. To start with, our recapitulating what we have passed since last 3 years. In March 11, about 3 years back, COVID-19 was declared as a pandemic. Right now, probably it's an endemic status, although there is some amount of debate. Almost 7 million deaths. We have heavily depended on vaccine doses, and there is a significant amount of doses are out, but still we are in need of more vaccine. As I said, viral particles are round-shaped, having this core that is encapsulated by a coating that's made up of protein. And this is an actual picture obtained by electron microscopy. If I try to make a less sophisticated one, this is the core with the spike type of proteins coming out. Essentially, this is the biggest protein structure coming out of this core shell of this SARS-CoV-2 virus. And the genetic material is inside. The SARS-CoV-2 virus, the spike protein is shown over here, which has a receptor binding domain on the top. This colored shells, they are carbohydrates. They are covering and disguise it from our immune response. So that's what we know about this spike protein. The spike protein, this blue color part, is the receptor binding domain that gets attached to human cell. And once it gets attached to the human cell, the virus is able to transfer its genetic material to host cell and replicate. So we knew that this attachment of this receptor binding domain, which is a protein, to our host cell plays an important role. So if you look at the structure of this spike protein shows actually 3 almost equally equivalent subunits, so to say. And when we know the binding occurs between the human and angiotensin converting enzyme 2. So besides RBD, receptor binding domain, the other part that you should be looking and knowing more and hearing from me more is angiotensin converting enzyme 2 receptor, or ACE2. Now, there may be some confusion, and I'll try to clear about this at a later slide. Now, when this binding occurs, the transmission to human cell actually happens. So this crystal structure, meaning the structure of this bound protein, were available when we started looking at it first. What we observed that there are a bunch of disulfides, and we knew from our chemistry knowledge that sulfhydryl groups or thiolate groups are oxidized to form disulfides. And this happens during the oxidative stress. So in essentially every time in our body, there's a balance that exists between the disulfides and the thiolates. So a question at a very preliminary stage, we started kind of asking, is there a redox connection to COVID-19 disease? What is the impact of oxidative stress? Started kind of looking a little bit deeper. We came to, we knew that there are antioxidants. We know that, having vitamin C is good for our body. Drinking colored, maybe, some antioxidant containing foods are good for us. We also know that oxidants are bad. There are radicals, and some of them are having a lot of oxygen. They are called reactive oxygenated species. There are similar to reactive oxygenated species, there are reactive nitrogen containing species called RNS. So a balance between the two types of chemicals in our body actually holds the clue to oxidative stress. If we look back in the thermodynamics part, measuring how favorable the thiol to disulfide conversion, we see that it's immensely favorable in terms of energy, gives free energy. There's a 15 kcal per mole gain of energy when a thiolate group is converted to disulfides. Now, we also kind of found out that there are some enzymes which triggers or which are overexpressed during the time of oxidative stress and keep upregulating or become upregulated during the oxidative stress, making this conversion. So finally, we understood that this is the part that we should be looking into, the role of this thiol disulfide balance on the binding of the SARD or the RBD part of the COV2 and the ACE2 receptor, the human ACE2 receptor that we talked about. And also in parallel, we try to kind of review more about the oxidative stress and their connection or its connection to COVID-19. So we did literature study, simulation at the level of atoms, and finally analyzing the simulation results. So why do we need supercomputers? I try to kind of think about how exactly we can tell to people who doesn't know about supercomputers. So I'm trying to kind of make an effort. So first of all, how big are this system, if we can answer that, and how many number of atoms or items we need to count? I start from looking at the average molecular weight of the protein as well as each amino acid. That gives me an idea how many amino acids in a protein we have. Average number of amino acids in a protein, about 422. If I convert that to atoms, mean atom number in an amino acid is 19.2. Again, the statistics is available in the web. So the average atom number in a protein is greater than 8,000. If I try to take number of bonds, another 5,000, bond angle, another 4,000, and we have a lot of water molecules because we need to kind of simulate these protein systems inside water. So all these things are basically informations, and we need to have supercomputer. We cannot use a desktop computer to handle this. There's a second element. That's time scale of the event. We are trying to simulate an event, not just a particular small part. So what is the time scale of this event? How fast atoms move? If we look at protein synthesis, that takes a very long time, 1 second. Go down to enzyme catalysis, this is about milliseconds. DNA unfolding, microseconds. Protein motions, a part of the protein dynamics that I had shown, about nanosecond. But we cannot stop there. If we look at bond vibrations, the atoms are kind of moving back and forth like a spring. That's about picoseconds, and electronic rearrangement is femtosecond or more. So if we think about, we like to see how the atoms are moving and that is giving rise to a protein motion, we'll have to have 1 million of snapshots to combine in order to understand what is going on. That means a dynamic information processing, and we need supercomputers for that. So the rest part is method. We set up the system. We download this from a collaboratory site. We add water. We start kind of identifying the sizes of this and then go into the simulation. We apply Newtonian mechanics, which can produce each of these atoms' motion very accurately. So from the results, we can understand what kind of interactions are happening between the blue part, which is the receptor binding domain coming from the COVID, and the human receptor ACE2 part. So we look at this binding region and understand different types of chemical interactions and deconvolute them to trying to predict whether this binding is strong or weak. So that's the analysis part we did. So the first of all, our students did a lot of literature review, and they found something really, really interesting. The angiotensin is a peptide which cleaves into angiotensin 2, 1 and 2. Very similar name. We won't probably understand that much unless I say that this molecule is bad for us. Why? Because this is an oxidative stress enhancer. It produces oxidative stress in our body. Why? We have a real good player in our body, that's ACE2 that I talked about that COVID-19 binds. What does that do? It breaks down this angiotensin 2 to some other compounds which are not at all responsible for oxidative stress. They are cool, they are nice. This is angiotensin 2 that actually restricts our artery wall and creates hypertension in our cells. So angiotensin 2 is really bad. And think about that. What happens when ACE2 is stopped its own action? That means angiotensin 2 will remain angiotensin 2 and oxidative stress will go on increasing. So that was what we published during the end part of 2020 by our students summarizing their finding, the main of which I'm showing it over here. Then we also, actually computer related simulation, we wanted to focus on the binding region. As I said, we noticed that there are two helices that kind of that goes inside the carb of this SARS-CoV-2, and the SARS-CoV-2, if it is tightly bound, it just moves sidewise in a direction showed by this green arrow. So the tighter binding is observed when these two long helices goes into this carb site. So we actually focus that from our analysis. Now I'll tell you two different computer experiments we did. The first one was to remove all different disulfides from both RBD and ACE2. By doing that, we allowed them to evolve by this molecular dynamics simulation, and we obtained the results. We looked at different binding. Now, if you think about, I have shown different colors. The colors which are green, meaning it is not very dynamic. The red part is dynamic. So we compared side by side, one where these disulfide bonds were not broken. That's the left-hand side. And the other, the disulfides groups, all of them are broken, which is the right-hand side. We saw that the binding part, this part has become extremely dynamic, as well as the part that was very far from the ACE2. So ACE2 is this part, the SARS-CoV-2 RBD receptor binding domain is on the top part. So we found this important distinction between these two, and later on our binding energy calculation did produce some significant difference between the binding of these two and that two. So we published that in at a very preliminary data. After obtaining this, we published this in 2020. Then in the following part, we try to expand this idea. And one of the really important experiments that we did was to remove all 4 disulfides. So this receptor binding domain, that's a part of the COVID or SARS-CoV-2, we removed all those disulfides and converted that to thiol. So basically reduce that. And we try to kind of see what happens to this small fragment of this of this SARS-CoV-2, which is responsible for binding to the human receptor. That produces something remarkable result. If you start from this structure, as you go through simulation in water, you'll find that after 100 nanoseconds, there is hardly any change, but at 200 nanoseconds, it started changing, but at 200 nanoseconds, the curve kind of area which binds tightly on the long helix of AC2 has completely changed. And in 300 nanoseconds, it kind of stays all through the remaining 100 nanosecond time and never went back to this. So from this, we found that the SARS-CoV-2 RBD receptor binding domain has a very important distinction or a very important conformational change if there is no oxidative stress, if there is no disulfides present, which on the other hand, if you try to kind of explain during the oxidative stress when the disulfides are really giving this curve kind of structure or binding site, curve structure of the binding site, that means that oxidative stress is essential for the SARS-CoV-2 to have a strong binding affinity to the SE2. Now, if we understand this, now this makes quite sense. It actually creates a vicious cycle. That's the thing that I wanted to kind of trying to kind of say here, that we know that angiotensin 2 is a major major bad player, or we can say that it is its property is to go to the artery wall. Vasoconstrictor is the right word. So angiotensin 2 is vasoconstrictor. It creates hypertension, and ACE2 cleaves this angiotensin 2 to form angiotensin 1 and 7. Now, if COVID-19 binds because of the oxidative stress, it keep on increasing the oxidative stress. And what we have found out from our computational study, that increased oxidative stress will keep the binding affinity strong. So it will go on increasing more and more, binding at a very short stage, and that we can call probably, the viral load will be higher during the oxidative stress, and this will create a vicious cycle of, oxidative stress as well as more virus particle binding. So we also found that, I'll stop my part of it, and we will show you some of the other research groups who have worked on this. So one group actually provided an experimental proof of this concept, the disruption of disulfides within RBD and SARS-CoV-2 spike protein prevents fusion and represents a target for viral entry. That's exactly what we are talking about. Then the antioxidation or lack of oxidative stress and COVID-19, there has been a review that also published in 2021. The Redox biology people found something interesting. There's an age dependency on the thiol disulfide balance. What does it mean? Well, if you remember, at a very early stage of COVID-19 propagation, we are thinking that children are probably not going to be affected. We found that the prevalent infection or the severity occurred in older age people, right? So the age dependency might be able to explain that. This group has found that there's a age dependency. Finally, there are some drug design people who have also used different thiol compounds which could target the SARS-CoV-2 inhibition. And these two are kind of proving that what we are thinking about is in the right direction. Well, I would conclude probably by stating again, the reduction of all disulfides into thiolate groups completely impair the binding property of this SARS-CoV-2. We also found that if you only reduce the receptor binding domain, it completely alters its structure and makes it incapable to bind the ACE2. Now, ACE2 being the regulator of oxidative stress, which I have said many times, its inhibition by RBD of CoV-2 increases the oxidative stress in a way that enables stronger binding of the virus particle, creating a cycle which goes on and on. The future direction, of course, we are trying to see if naturally occurring small molecules can act as a reductant to this disulfide. So that part is still ongoing in our lab, and we'll focus on some of the new drugs, thiolate drugs that has come out after this. Finally, I'd like to acknowledge the collaborators. Carl was one of the persons who represented this work to WISES and got a first prize in Wisconsin-wide WISES competition. That's called Quick Pitch, right? Quick Pitch competition. Alex is over here too, who has contributed, both of them contributed significantly on the computational experiment paper. Sanjay, who has joined a medical school, and Jonathan, who is working as a theoretical chemistry group in U of M, University of Minnesota, worked on the review part, literature review part, along with Carl Fossum and others. Harrison and Bethany was also part of this entire project. I would like to thank Sanchita, who worked with this group all the time, and especially she has been instrumental in getting the theoretical review paper done pretty within short time during the summertime, especially. Finally, I'd like to acknowledge ORSP for organizing this event, the Blue Gold Center for High Performance Computing, especially the system administrator, thanks Taylor, who is phenomenal during the time of COVID-19. We got a lot of help. UWEC for my sabbatical leave during the time of this first part. I was still in sabbatical, so that enabled me to kind of do some really quick study. The first study was done almost like in 1 month of time. I'd like to thank NIH and all the researchers who made available the structural information for this computational tools. And I'd like to thank you, all of you who are here and who are in Zoom. I'd be ready for answering any questions you have. Questions? Is there a way I can see? Yeah.
I've been told also that you need to repeat what I'm saying because
Sure.
I'm not gonna pick up by the microphone.
Yeah.
Can you give me just a rough outline of the computational tools that you've used for this? Because you're talking about huge numbers of particles and understanding their motion at different levels of theory. It seems like a huge task. Is it one kind of software, or is it a lot of different approaches?
Sure. So the question is, what kind of software and computational experimental tools that I have used during the simulation? The first simulation, or the first tool for simulation, was a classical molecular dynamics simulator. That's a single program. It's made free for academicians by University of Illinois Urbana-Champaign. The name of the program is NAMD, Nanoscale Molecular Dynamics Simulation. The other part that we have used to deconvolute the energy difference between the two, and that is a separate program. I think we got it also free from another part. That requires some, a different type of calculation based on charges. But this is also available. The name of the program that we have used, APBS, the Poisson-Boltzmann calculator, which calculates the energy of a protein molecule based on the charges. So different conformational or conformational ensemble that we got from this dynamic simulation. We ran this APBS, which gave us a large number of, quantities. We took the average of that in each case and provided a number.
So the theory is understood to be accurate, like the machinery that's behind these programs, that's been well benchmarked, I'm guessing. And if you assume that, then what you're really testing is your model here, like your proposed mechanism.
Yes.
And it seems like it passed the test.
Yeah. So the question is that if these kind of tools are already benchmarked, then we are only testing the already standardized tool to understand the biophysical aspect of this. And that's true. We did not optimize the methods at all. We took the optimized method and applied on this system because we had little time, we can't have a unstandardized and then standardize and get it, but rather we took a short approach of using standardized tool to look into the questions that we are trying to answer.
And the experimental follow-up work that you cited, that's pretty awesome. I mean, that must have been pretty exciting. Do you know this, folks?
No. So the question is the experimental part that we had shown later was, do we know them? No, we don't know them, but I have been able to review one of their work. All of them have actually cited our work. There's a number of citation for the review work, which has crossed probably 200. And the first paper has about 140 or something like that. So a lot of people, but some of them are good work, and I took some of this which are probably meaningful and will have some other important parts or other important aspects as well. Yeah.
Go ahead and ask your question. Go ahead. One of your earlier slides has something about machine learning or deep learning. I can't remember which it was. Could you just say a little bit about that? I suppose it's under the hood of the.
Okay. So the question is, one of the slides had some machine learning. Can I talk a little bit about that or not? I think you're talking about this.
First one.
Yeah. Thank you for that question. Yes. So drug design and the application of machine learning is kind of new concept. Machine learning has been used for predicting a lot of things, right? From image to, cancer 4 years from when the image has been taken. So we're exploring this part, which utilizes deep learning, meaning deep neural network is sort of part of the neural network technique, and applied to, finding out, predicting which molecule can bind to a arbitrary protein. What we do, we have, over the time, we have a very large number of database of small organic molecules. We take those small organic molecules and make a high throughput docking study on this. That docking study produces some numbers. That's the actual, Gibbs binding free energy type of analog. Those numbers are input one. Input two, we use something fingerprint, which converts a small molecule into a basically binary number. So that's Morgan fingerprint and the Gibbs binding free energy are the two types of input fed to this neural network at the same time. And over the number of iterations, there are some retentions and there are some, discarding, training, and testing and validation. These three procedures are repeated. And what we are trying to do, I mean, this takes a very long time, and I don't feel I'm comfortable to say that we are helped by this. This is the deep neural network-based technique that I borrowed from a 2020 publication that has happened recently from another group. And we are trying to see if adding quantum chemistry information, could that be speeding up the machine learning or not? Meaning giving more accurate description of the binding free energy, so to say. So that's the part that we are still working on.
Yeah, we got a couple of questions from online. So the first one is, I'm curious if your research has involved looking at the variant strains of COVID or if there would be any difference in doing so.
Well, we did actually look at different variant strains, and by using our biophysical chemistry group in 2021, we found utilizing the same technique that Delta and Omicron binds more tighter than Alpha, Beta, and Gamma. Alpha, Beta, and Gamma. Wild type and Delta and Omicron were very, okay, sorry. The question is, have we seen the different variants using the same technique? The answer is yes. And there is a publication already in the web.
And then the next question is, it seems as though computational work lends itself to remote learning forced by COVID. Was it easy to involve students remotely or any drawbacks and what that looked like during COVID?
Well, remote learning is always difficult, but I think through this process, so the question is, sorry, I'm forgetting to kind of repeat the question. The question is, during the COVID-19 time, we did this, so that is a kind of remote learning. Was it difficult? We all kind of evolved, and if I do remote learning now, I'll be far better than what I was experiencing at that time. But thanks to our students, they were very eager, helpful, and they all kind of took the leadership in different frontiers to kind of make this happen. And I'm simply thankful to all the students here. Any other question? Yeah.
Is there any evidence that antioxidants have effect on COVID binding?
There are, but I don't think the clinical trials, there may not be many clinical trials. So the question is, are there any evidence that antioxidants are actually working to COVID-19, to reduce COVID-19 severity or not? I'll have to check, but I don't think clinical trials have been conducted on in an unbiased way, because this experiments, clinical experiments, are difficult to actually conduct. How do you do? Unless you have COVID-19 cases, then you'll have to kind of find out how many of them have actually taken regularly some antioxidants, and all these monitoring and other things are extremely difficult. So I don't think clinical trial exists.